before we get into the specifics of our research. See my eyes here, I do vision work. First with the middle of the nose. So as we start, I do visual neuroscience, so my big focus really is neuroscience. So I want you to think about the brain for a second. This is a 3-pound mass of like jelly. It's like a bag of tofu. But it's what we can use to think about ourselves, to think about the stars, to interpret the whole world around us, and to interact with everybody. So to me, it's really what I call the crown jewel of the human body. I use people who like study the kidney and love the kidney, but personally, I love the brain. The brain is what makes us us. Now, that leads to questions of how does it work? How is it organized? And as soon as we get into fields of neuroimaging, like PET imaging and functional MRI, things that tell us about activity in the brain, people got very, very interested in defining what different regions of the brain did, what these different segments did. And one area that's been very well studied, because it has a history of studies in cat and monkey that's very extensive, is the visual parts of the brain. And so my work is in visual neuroscience, and I actually really like it because of this large foundation, because I can take this in as a branching point and look at lots and lots of different questions about the brain, everywhere from things that are how is this organized fundamentally, to how does the brain change in disease and damage. So today I'm going to take you through a little bit about vision, and then something about our methods. We have some specialized methods that give us a very, very good way to map out parts of the brain, something that some people are lacking in this field. I think that we can look for if you're reading brain imaging literature in the future. It's kind of my goal. Brain imaging literature, this is bad, this looks better. So we'll talk about that a little bit. And then I'll take you through what we call the PRISM project as an example of how we use our methods, and you saw some people you heard earlier walking around the videos of that, so that kind of stuff. Okay, got my. So, vision is more than what your eyeball sees. Your brain is integral in interpreting this. If you've seen this illusion before, don't yell out the answer. But just to give you an example of how important your brain is in making decisions about the world around you, and how perception is more than just the photons hitting the back of your eye at the retina. I'm going to show you some visual illusions first. So, are these just disconnected lines? Phrase it a different way. Start trying to second guess me. So, does this look like a set of disconnected lines to you?
Yes.
Yes. Does anybody see anything greater within these lines? Raise your hand. Jacob's seen this. What if I tell you, can you see a letter? Yeah. Does anybody see a letter in here? How many people see a letter now? Oh. Yeah. Something you can't unsee, which is why I tell you guys, if you've seen it before, don't shout out. It's like, oh yeah! So it looks like the letter E. So it's a capital letter E like this. See that? So all that your eyeball is seeing, all that's hitting your retina, are information about these black lines on a white background. When I first saw this, I was like, are they chairs? Are they steps? And now they're an E, and your brain can use the shadow information to overlay your perception of letter E on this. So this was fascinating. Your brain is completely constructing this. So reality is what you perceive, really, not necessarily what's really directly out there in terms of energy in the world. So I'm going to show you a couple more complicated examples of this. I'll stand up here so I can see what we're looking at as well. So these are two tables, and other than me showing illusions, how many of you would believe me if I told you that these two tables are actually the same size and shape? My granddaughter might prove this to you in a minute, but how many of these actually look like that to you? Red and green tables, same size and shape. They don't, right? Just not at all. Okay. We're going to draw some lines across them. And we're going to move the tables over here and take these lines, rotate them, and move them over. So what your brain has done is reinterpret the size and shape of these tables with depth. So back this up. So what the red table is, it is the green table. It is rotated on its side to be the red square here, the red looking square here. This is the same blue line as this line. They're the same length. This yellow line is the same length as this line right here. Now if we look at it again, I take this overlay of lines, turn them, they're not stretched at all, and move them over the overlay perfectly. So as you developed, they really do, there's no stretching. People don't believe me, they've actually come up and measured it before. So they really, really do match. What your brain has done is taken your experience with objects in depth and size and shapes of objects and learned to expect how things look with different perspectives. And because of the lack of other types of depth cues in here, this looks more like a squarish red table, and this looks like a rectangular table here. And your brain is overlaying a perception that overrides the actual physical stimulus coming in. So we have not only the ability to like put together just the line information, but make more complicated judgments now about how these things look in depth. We can get even more complicated and start making expectations and guesses about how things look in 3 dimensions. Things like face. So we're moving up basically the visual processing system. We're going from lines to things in depth to things in 3 dimensions like faces that we give meaning to and have specialized processing pathways for. Now this is a mask, a hollow mask. This is called the hollow mask illusion. This mask is hollow on the inside and is going to rotate the same direction for the whole time I show you the video. Watch and see what it looks like to you. Oops, not like that. Click. Hang on a sec. So the mask is rotating around very slowly. And you can kind of see the facial features of the mask, out of 3 dimensions here. As it rotates around further, you're going to look into the mask, and now it looks like it's rotating the opposite direction. And looking out at you. You're looking into the mask in depth. But your brain is so used to understanding faces of a 3-dimensional object projecting towards you that it overrides this image and makes you feel like you see this as a mask looking out at you from the screen. We can have, I think, goes around one more time. Because you're like, no, do it again, watch it really do it. So watch again, so the mask is sticking out, and as you see just enough internal information, it looks like the head rotates the opposite direction now, and the face popping out at you. It's still rotating this direction, but it does not look like that. And this is your brain overlying perceptual information onto that mask again. Okay, so I see these things and say, wow, this is cool, I need to study this thing. So we go to a much more fundamental level to start understanding some of these, but this is why I personally like looking at things like vision in the brain and trying to see how these perceptual bits of information are built up. Okay, so stepping back from illusions, we have to talk a little bit about the visual fields and visual maps. And I look, like I said, at very fundamental pathways of the brain to start, and then you can apply these to many things. And this means I have to see how the incoming visual information gets into the brain. And that comes in through the eyeball. When we talk about things coming into the eyeball in terms of visual space, imagine this is a circle of visual space with your eyeballs focusing right here in the center dot. We call this your visual field. Now, we divide that visual space into what's called a vertical meridian, so this vertical line, and a horizontal meridian, horizontal here, and your fixation point. And your eyeball, this is the cross-section of your eyeball, has information coming in through the pupil, goes to the lens, and falls in the back here on the retina. And as your eyes are still, all the visual space is in a very ordered manner, coming in from visual space, falling in an orderly fashion on the back of the eyeball. Now this orderly information is called a retinotopic organization. It's on the retina, it's organized across the retina, and as this pattern of organization enters the brain, it stays organized in retinotopic coordinates. So it stays organized as a retinotopic retinal space, kind of visual space to retinal space, and that retinal space is maintained in the brain. And we can actually measure that. And we can measure this then to help parcel it out different regions of the brain. Here is a horizontal section of the brain, looking at the eyeballs and how things get back to the brain. And something really important to this that I want to tell you about today a little bit is that information from each half of the retina goes to the opposite half of the brain. So that means all the information in visual space on the left side of the world goes to the right side of the brain. And the right side of the world goes to the left side of the brain. It only gets integrated much, much higher up in the processing pathways. So all of your initial information coming in goes to opposite sides of the brain from this side of the world. If you've got brain damage on one side of the brain, you lose vision on that opposite side of the world. So it's called contralateral side of the world. Sorry? Contralateral side of the world. Ipsilateral side of the world means on the same side. So we talked about things going from a contralateral side to an ipsilateral side. And visual cortex is all primarily in the very back of the brain back here. Kind of a 3D shape, we have image coming into the eye, coming through the lateral geniculate nucleus, this little part down here. It's a very specialized part of a below internal brain structure called the subcortical structure that acts as a sensory relay station for almost all of your senses. So information vision comes into here, maintains this organization that follows the retina, and then is sent back to the back side of the brain. And then we have repeating maps, organized maps of visual space across visual cortex. And with our methods, we can actually measure these. So here's where I want you to think about neuroimaging in the future. So neuroimaging looks at activity in the brain. And in many cases, you can say it has a spot on the brain. You get a spotted activity like, hey, faces make this spot, the key make this spot in the brain, hey, this is part of the brain that faces giant big area of activation. We can actually measure out individual regions of visual space with the idea that each map of visual space can do a specific computation on that space. So it can look at color, it can look at processing lines, the face region itself may be organized into multiple maps of visual space. We can get a very, very careful, detailed measurement of the brain that we trust very much and look at other information on how the activity inside that brain also functions. So we have kind of an idea of having a map of visual space, and that map does a particular thing in vision. We can look at how these maps are organized, and therefore how these computations or processing power happens across the brain. So in my lab, we look at several different things. We look at how these representations are organized across the brain. We can look at what their functions are, so looking to see then these patterns of general brain activity, how they fall across these maps of visual space, and kind of break up these big chunks of activity in the brain that people study. How we can play with these under normal conditions, which I'll show you about today, and then how they also change in disease. Say things like Alzheimer's disease, other things that cause decay of the brain, ways we can detect early disease using these, and ways to understand things like eye disease and vision loss. Okay. So I'm going to take you through our methods now, just to get a little brief idea of how we look at these things and then how we apply these in some cases. This is my brain. It's not generally this atrophy looking and smoothed out. This is our neuroimaging software that allows us to make 3D renderings of a piece of cortex. This is my left hemisphere. My eyeballs will sit right up here. This is the inner part, so the brain has two halves. This is the inner side, the ear would be end of the screen. And the visual information is organized at the back here. And based on measurements, I want to show you first how these early, our measurements, based on our measurements, how these early maps are organized, and everybody in our field agrees with these types of measurements. So these are, again, our visual field. This is the right half of the world, which goes to the left side of the brain. And you'll get a pattern of responses that are organized across the visual space overlaid on the brain here. So this is visual cortex back here, the area of the brain involved in vision. First visual area is named V1, which is very nice. Visual area 1, primary visual cortex. All the sensory areas is a number of 1. And we go up to like 12 and start naming things differently. So it gets to be kind of complicated. We're going to take you just through the first couple. And what this is the lower part of the world is up in this upper part of the brain here, progresses smoothly across to this upper part of the world down here. So every point in space is neatly organized across this map. Dotted line is the horizontal right in the middle here. The fovea, the central part of vision, which you use to focus and read with, is this yellow representation here, and it's quite expanded. So we devote a lot of our brain to dealing with all the information coming up from a very central vision, and a lot less to the periphery. And then we get pieces of maps that are organized around the central map. So you can kind of step out from the central V1 to the next areas, and then we get really complicated organizational patterns of how these maps form into kind of clusters of maps across the cortex. So we measure this, again, neuroimaging points. When you study neuroimaging, they do group studies. People average a lot of data together and can make certain claims about what their brains see. Now, others of us, like my lab, look at these in individual subjects. And for visual field maps, you can't average together this information. It's very, very specialized. It's very hard to get information, and it differs across individuals. So the size of the first visual area itself differs by a factor of 3 across people, which means it moves everything else in the brain around in different spots. People average, they guess where in the brain they're averaging across. They guess what an anatomical point they're at, just average the data across there and look at that, which loses all of our information that we use in our methods. Some people have really big visual cortex. People's brain sizes overall, though, don't alter with the size of different subdivisions. So some women have a giant visual cortex. They're smaller somewhere else in the brain. My advisor in grad school had a really, really big V1, and he was all excited about this until we asked him where it came from, and suggested it might be his frontal lobes, which did a lot of thinking power. He was not as thrilled about this, but darn, V1's really big, something missing. So his brain wasn't big overall. It's not that the brains are scaling up, but the different brains differ in how they're organized exactly. They all have the parts in most cases, but the way they're set up is different, and the sizes are different as well. So we look at individual brains really carefully. This you can see my brain today. So this is my eyeball. This is my nasal cavity, nose down here. Skulls on the outside here. And what we do is look really carefully at just the cortical sheet of gray matter surrounding the brain. So where the cell bodies are in the brain. This is where all the computations are done. We do this by kind of coloring in. Jacob and Mark get to color. Glenn get to color in my lab. They get to color all this purple in and basically define where the edge of white matter, which is the kind of connections between the parts of the brain, where that differs from the gray matter. And then we take all of that cortical sheet information and we can look at it. And so this cortical sheet, imagine, is like one giant connecting sheet. It's like crumpled up like a ball of paper and it's fit inside your skull. And we want information that's spread across the sheet, not clumped into spots of activity, but flattened out to look at the information of these visual maps on this sheet of cortex and how these maps change. So we can draw it like this, coloring. We can make the same 3D rendering and label different anatomical parts of the brain. We can blow it up in these special algorithms, take this estimate of the cortical sheet, render it into 3 dimensions, and flatten it so you can just label the valleys as sulcized dark and the gyri or peaks as light gray, and then we can look at data on top of this. We can also take this whole sheet and flatten it down and look for these patterns of responses that look like maps of visual space across them. So this is a point right here, flattened all out, and you know where all these labels are, how the brain can be organized. Okay, so just a couple things about methods, and I'll show you how we use them. This is an MRI scanner. Now MRI gives us that structural image of the brain, the picture of the brain where I showed you my eyeball. fMRI, functional MRI, can tell you about how active different regions of the brain are. It does this by looking at blood flow that's correlated with neural activity. So an area that's more active will have a higher signal in fMRI. And we can drive this signal in visual cortex with certain types of stimuli. And the field standard is called the Chiltern wave method. We use these really, really fun stimuli of black and white flickering checkerboards that we stare at for like an hour. And these map out visual space in very particular ways. So these are flashing, moving checkerboards. Primary visual cortex goes, oh my god, there's black and white edges, it's really exciting, it's really, really active. And we use what's called a periodic stimulus that moves smoothly through visual space and repeats over and over again. So this is a rotating wedge that rotates around and around and around. And each region of cortex that is organized into a map of visual space has one neuron that likes its point in space, and the next neuron overlikes the next point in space, and the next neuron overlikes the next point in space. And so you can see a sequential activation of each chunk of cortex that corresponds to the periodic movement of the stimulus. So we have, this is a plot of one little point coming up and down here across the brain. So we have a point that goes, oh, stimulus, no stimulus, oh, I'm active, oh, I'm not active, oh, I'm active, oh, I'm not active. This is the traveling wave, this wave of activity that moves through cortex that corresponds to the pattern of map of visual space. So you have a wave of activity that says, oh, I like the fovea, I like the periphery. A wave of activity moves along here. It's not what to do. Sneaky button on my new pointer. Okay. So this pattern of activity lets us color code space. And to look at a map, we need 2 pieces of information. We need both how it changes around the angle, how it represents that, and how it looks at the very center or foveal part of the space out to the periphery. So we use 2 different types of stimuli for this. One is the rotating wedge we just saw. When we color code visual space, so we have that vertical part here, and the upper part in cyan, down to the horizontal is bluish purple, down to reddish orange. And we can then overlay, we use these are called pseudo-color maps that tell us the activity in the brain, how it corresponds to visual space, how these colors represent different parts of visual space activity. So in a cartoon, our first visual area will be neatly organized like visual space, running from the cyan all the way up to the reddish orange. And here is a cropped down part of the left hemisphere looking just at that part. And this is real brain activity. It's color coded to represent the parts of space that the stimulus was in when that area of cortex was active. So when the stimulus was up here, this part of the brain was active, and we color coded that cyan to that part of space. And we can march across space from cyan to blue to reddish orange, and we can see some reversals jump back. So we can use this to define each part of each map. But we only know how far the map runs this direction, not anything about this direction. So this is called the polar angle. The next map is called eccentricity. For this one, we still use checkerboards, but we start with a ring in the center that expands out and starts over and over again, repeats over and over, and we average together all these cycles, like this wave of activity that happen here. We now color code space with Lily Rainbow, starting from red in central fovea out to purple in the periphery. Our cartoon shows U1 having a map that's now this color orthogonal to the polar angle map, perpendicular to the polar angle map. So these 2 maps let us uniquely identify each point in cortex that corresponds to a particular point in visual space. So this map now is red in the fovea out to the periphery. It means when we look back here, all of these like the vertical meridian, all the neurons along this region. Oops. But these neurons like the vertical meridian in the fovea, and these neurons like the vertical meridian in the periphery. So we can see each of these pieces of cortex is a unique part of visual space that it likes. And these maps change as we get into higher heart rate processing, which is less regular information about the visual world as we get, say, looking at faces, but we still can find these maps in our data. So we have some nice definitions of that. I won't take you through all the details, but you can see we go back to cyan, we go back to purplish and back to cyan. These are ways we define how these maps differ. Each map in visual cortex can only correspond to one representation of visual space. We measure these things with what's called coherence. And this is a way of statistically looking at what a significant signal is in the brain. So things above a certain cutoff threshold, we call them significant. We take these as actual data. Things below that just act counted noise. So our lab and a few others have now used a new method as well. This is my last bit of methods, so hang in there until I get some people staggering around the world with prism goggles on. So these methods are called population receptive field modeling. So in humans, we have not been able to go with an electrode because we're not allowed to and it's not safe. Kind of reverse reason, I guess. But it's not safe and therefore we're not allowed to stick electrodes in human heads unless they're under surgery to measure out responses like we do, say, in monkey and cat for all the visual literature is. Our neuroimaging has gotten really, really good at looking down to what we call a voxel, which is a volumetric pixel. It's a little chunk of space, and you look at all the activity within that chunk of space. We're on the order of hundreds of thousands of neurons within that chunk of space. And that's at our best. It would be lovely to know more information about that voxel than just where it represents in space. What if we could tell, on average, what the receptive fields of these neurons are? What the properties are of the responses of these neurons to points in visual space? So we know where they're like, the most preferred center of visual space can be told to us by the traveling wave method. But how wide does that neuron actually correspond to visual space? Some neurons like to stick at just a little tiny part of the world, and some neurons like to look at a big expanse of the world. Things like V1, which is the first part of visual cortex, like to have a really, really accurate representation of the world. So they have very, very small receptive fields we think in monkey. We know in monkey, we think in human. Now as you go up to maybe face processing, where you want to know information across the whole scene to understand what's going on, those neurons still have a preferred center of space in many cases, but they also want to look across the whole scene to have a really broad receptive field. So everything that it covers, all parts of visual space that the neuron covers its receptive field, and that receptive field then has a preferred center. So previous methods can only look at the preferred centers. But these methods allow us to look at the spread of the responses as well. So this is a big step forward in what we can do. So we can look to see exactly how the receptive fields match monkey in terms of human measurements, and also how they change in disease and other types of studies we'll talk about in a bit here. And this is something we haven't been able to do before. This approach can take any type of stimulus, which is these guys as well, or these bars down here, that moves across visual space in a regular manner. And it estimates out what you'd expect to have as a response from that voxel based on any parameters of receptive fields. And we do an iterative approach and just keep checking and checking and checking and get the best fit for what our receptive fields could be. And basically, our computer can tell it to go, it runs our algorithm all night, and it gives it information out that tells us not only what part of visual space this neuron responds to, but also how broad that response is, and how it can change in shape and size and location across different types of studies. So we estimate receptive field size, we put several parameters in, and we get some data back out. For those of you who haven't seen the scanner before, subjects lie in here, and then a mirror can view a stimulus on a screen from inside the bore of the magnets. They're lying in here, surrounded by an MRI machine, and you have a projection screen that comes in with the stimulus information you're staring at during these studies. And a thing to note, during all of these studies, I talked about the fixation point in the center visual field. This only works to measure these maps if the eyes are still, because you want visual space to have a static image on the back of the retina, which then sends that information to the cortex. So you move your eyes all around, your brain's getting information about all different parts of visual space, and you're getting different parts of the stimulus measuring it. So we keep very still for a very long time staring at checkerboards. And now, having done this for years, I walk around and I see checkerboard patterns that all look like they're moving to the side. Like I'm fundamentally changing my brain by staring at checkerboards too much, but they are quite useful stimuli for us. Okay. Just a couple more things on the model. People talk about receptive fields while looking to see, like I said, where the preferred center is and the width of the receptive field. This is an example of V1, that first area of visual cortex. A neuron likes a little itty-bitty bit of visual space, and the traveling wave method is great for measuring this. But it's not great for measuring things that are really wide. So this is object cortex. It's looking at really wide responses. And these are parts of the visual cortex that do things like, hey, I'll turn on because something's happening visually. And they have a little tiny peak change as you step through different parts of visual space. And the newer method lets us measure this little tiny, tiny peak change on top of this baseline. Oh my God, there's something visual. So turns on to vision, also gets a peak change across cortex. So, and then we can get size estimates, like I said, of how these change. Things that are important to think about is the sizes of neural responses across the map change depending on what part of visual space it represents. So if they like phobia, they tend to have very small, this is the size up here, center to periphery out here, very small visual fields down here. They want really high acuity vision, you want to be able to read, you want to be able to see things in front of you. And as you go out to the periphery, they get bigger and bigger. Okay. So we use these methods to look at patterns of all these maps across cortex, like step one. So step one, we found that cortex is organized not just into a few visual field maps, but into this pattern of clusters. Clusters of maps that look like they come up in development and differ across evolution in terms of the numbers of clusters different types of animals have. And we think that we have a gene that may code for having a new cluster of map that pops up with a new set of computations. All these maps have similar properties in terms of receptive field sizes, similar computations. They may all do motion processing, they may all do color processing. I'm going to show you, this is, again, our left hemisphere here. This is the back part, eyeballs will be up here. You guys know much about the brain, this is temporal lobe coming down here. This is the first view you saw with eyeballs up here. It's kind of the other surface of the side of the brain. Same hemisphere, just two different angles. And you can step out maps all across this part of the brain. So all these colors of activity are going to be this eccentricity map, the center to periphery organizational pattern, and you can see how much the brain starts getting covered by this vision. Very basic vision, actually. So that's object cortex, motion cortex, more motion, other types of motion cortex. Coming up to what I'll talk about today, which is looking at visual spatial information. Primary and early processing of form information, and more color and object information. So this is really basic color. That's all very basic vision. That's all very organized into these visual field maps. This is the human brain. We end up covering almost 20% of the human brain with just visual processing alone. So we're very visual animals. And of that part of visual cortex, a significant, if not all of it, is organized into organized representation of visual space. So it's a very fundamental measurement that lets us look at details and patterns across much of this brain. Okay, so that's it for methods today. That's what we're doing in measuring our brain. But we then can apply these methods to many, many different types of measurements. So like I said, we can measure the organizational pattern I just showed you. We can look within those maps then at things like working memory. How do you hold information about visual space in memory over the short term? We can do studies where we look at, now, really exciting three-dimensional cubes. We've stepped up from vector. Three-dimensional cubes, you have to remember information in a brief flash of what each of these targets looks like. So this can drive certain parts of the visual cortex. It tells information about how we process short-term memory within these visual field maps in certain regions of the brain. We look at things in the dark, where we have really, really low, like starlight conditions, and look to see how the brain changes under those conditions. The retina is set up so there's some cells that deal better with highlight, called cones, and some cells that deal better in low light, called rods. And in low light, you get different perceptual effects where your brain, again, fills in missing information, kind of like your blind spot in your retina. Here we're filling in information in really low light conditions and night vision. Brain kind of fills in stuff that's missing. We do something called diffusion tensor imaging, which is a different type of neuroimaging, looking at the structure of white matter connections. So you can look to see how these different visual processing pathways are connected up across the brain. My lab's looking at things like Alzheimer's disease to see how these areas change early on in Alzheimer's. So it turns out that Alzheimer's disease has problems with visual spatial processing and other visual deficits that are often overlooked because the memory deficits are so severe. But early on, patients tend to start seeing the eye doctor saying, I have problems with vision or problems understanding this, and it doesn't get picked up until later on. And we find that these visual field maps show a lot of problems. They get really disorganized and get holes in the responses really, really early in the disease. And they might be able to use as a marker to differentiate what's happening in many patients. And then we're looking at things like auditory cortex and genetics and robots, apparently, to look at how these things work under different studies as well. So, but one of my favorite studies to talk about is looking at how things can be altered in normal subjects. So we look at fundamental measurements in normals. We look at normal measurements in disease and disordered populations, with people with lesion studies and strokes as well. And we also take undergrads and grad students and change their brains. So this is one of those studies. I love to look forward in the front row. You all volunteer today. No, just kidding. Very careful volunteers. So we know during development that the brain, especially the visual cortex, is quite able to change and adapt to its environment, but only about to the age of 5. And after that, the brain shuts down, in the visual cortex at least, its ability to reorganize very much. So you have a stroke in the back of your brain. You don't get that function back. The brain doesn't regrow. There's a lot of research looking to see how we can fix these things, both in the spinal cord and in the brain itself. The central nervous system does not respond well as an adult to injuries. And so we're looking to see if we can change the brain in certain ways with extreme changes of sensory input. So we took normal adult humans and altered their visual input and looked to see what happened to their representation of visual field maps. So remember, the visual field maps had information to the right side of the brain going to the left side of the world, and the left side of the brain going to the right side of the world. Now what if you flip that? So you have goggles on that take your world and left-right reverse it. If I turn these left-right reverse, you have a problem with the visual motor integration. So as you want to reach to one part of the world, you would see this over here, but in reality, it would be back over here. So you'd be reaching to the wrong side of the world to be grabbing what you're seeing because your world's reversed. Now we know from behavioral studies since the 1800s that people can actually adapt to these changes in vision over several days. And if behaviorally people can learn over days to be able to reach to the correct spot in visual space when their vision is flipped, something must be changing in the brain. And so we wanted to look to see if we could find where these changes are happening in the visual motor pathway. So you can I think about the brain being a way to take in information about the environment and then making decisions on how to act on the environment. And so here we're taking in information with vision and making decisions about how to reach the world to interact with it. And we're screwing up that pathway fundamentally with some apparatus like this. So what would you do with world reverse? This is my grad student who normally can catch a football quite well, but he is wearing prism goggles that restrict his field of view down and have prisms inside which take all of this side of the world and make it look like it's over here, and this side of the world make it look like it's over here. And I will show you him trying to catch a football. And he knows it's backwards, but his brain is so ready to plan the way to catch this. He gets to be endlessly embarrassed by this for all future time, because that's just the classic failure of trying to catch a football. So he can catch a football just fine without these on, but he knows it's coming down the wrong side, and when he plans that motor trajectory to reach up and grab the football, he goes the wrong direction. He's like, coming down, oh, quick, grab it, and it comes over here behind him. What if I told you that I could make people after 4 days of writing these be able to move and catch the football correctly? Would you believe me? Yes. Talk on it. Seems quite crazy, right? So, what we did is take these custom-made prism goggles that you see here. Here's your prisms that are showing opposite sides of the world. We found really motivated subjects who decided to wear these goggles for 2 weeks continuously. So they wear these goggles night and day with no other vision at all. So only the goggles. They had like blackout goggles on for taking showers and walking around and had us help them around. They got nauseous. It's very, very disorienting. It turns out your vestibular system does a lot of stabilizing of your worldview, and so as you move through this, you are so screwed up in how you interpret your movements through the world that it feels like you're looking through a jittery camera. So when you look around the world, it looks very stable, you can tell what the horizon is, but if you're running with the camera to move through the world and watch the video back, the video looks like it's bouncing. Well, you put these on and the world immediately looks like it's bouncing. So it looks like you're running around with this crazy bouncing video. So people got quite nauseous in my study. I apparently make people throw up a lot of my studies. So this is one where they get really nauseous and dizzy. On arriving at the campus, the IRB was quite excited. Human subject use because everything threw up. They were all very, very motivated and good with it. So they put these on, and for about the first 4 days, they were not able to function very much at all. They did a study like this in monkey and saw some changes in the brain. The monkeys put these on and promptly stopped moving or eating or doing anything for a month. So they're very disoriented. I'm much more like a monkey. I put these goggles on and couldn't figure out where the door was in my office. I was like, oh my god. Other people put these on and walked along just fine. They looked at their feet and went, oh my god, it's the wrong foot, and promptly fell down. That was the guy's football. So he's actually pretty good in these and still can't catch football. But after 4 days of wearing this continuously, even in one subject who was more like me and couldn't function at all, they were able to move through the world with a lot more ease, and by the end of the time, perfectly fine with the goggles on. Lost the idea of what left and right was. So people got confused about, and they say turn left, and they went old left or new left, and they go, okay, which one was the old left? We get very, very confused like that. But we did daily behavioral measurements to see how people could train up on this visual motor skills. And we did about every 3 days functional imaging measurements of these visual field maps in a particular part of the brain, the part on this top part of the brain that deals with visual spatial information, we look to see if those maps change at all, because we think they're involved in the visual motor pathways. So we're saying that somewhere in the brain, we are remapping our information about where in visual space something is and where we're going to reach. And so we're looking to see if we can find where these things overlay. So I'll tell you first about this experiment. Then these brave souls returned several months later into the beginning. So I did it for another 4 days later on to see if we made some permanent changes in the brain, see if we could get the maps back again. This is our first subject, and she has just put the goggles on for her first day of experimenting. And she's now going to wear them for the next 2 weeks. And she's trying to stand up straight. And it's not going so well. She's supposed to stand straight and turn and walk to the door. And she has a walking stick right now to help her balance. Since she can't see her feet very well because the goggles stick out a little bit, so it's kind of helped her step. And so this is the horizon is going wildly back and forth when she moves her head on. I'll put these on and get my head crooked and spin kind of to the wall. They overcorrect the wrong direction. It gets kind of problematic. So they're trying to show her, here's your stick. Go forward toward the door. All right, she's still trying to figure out what the stick is. You can see the stick, and she's trying to like follow it like this. So people will get this down. Yeah, okay, so she's really trying to go forward. That's not working out so well. And she has to get to the scanner to take a picture. So this is on day 7. So that was day 1, she's trying to leave the lab. Day 7, she's trying to leave the lab again to walk a maze. This is track for 7 days. A little more stable. She has to touch all certain spots with her right hand only. Day 1, she didn't make it down the hall. She ended up on the ground throwing up, so didn't ever make it. So day 7, this is a vast improvement. But she said at this point she'd get up at night and her spatial map of her room would come backwards. Everyone had to have a caretaker at home to these sites too to make sure they were safe. So maze walking, I'll show you just quick behavior results here. In maze walking, in the early days, of our 3 subjects here, they went from being really, really quick and taking about 17 seconds to walk down the hall to taking indefinite to very long periods of time. Drops back down in terms of time until about day 10 when they get really close to baseline. And this peak is when they take the goggles off again. Oops. So, goggles on in pink, adapting over time, goggles off. So they've adapted. It takes about a day to go back to normal vision after they remove the goggles. So your brain has changed to deal with its reverse vision. And then we expected this to possibly happen for other behavioral stuff, but when you take the goggles back off again, it takes some time for the brain to readapt back to what normal vision was. So this is rather disturbing. Subjects expected it, but still took it off and went, oh my god, my eyes are backwards. And they had to keep moving their head and their eyes back and forth, trying to get how to look around. So this lasted just a couple hours, with most of it all within about 24 hours. But it's a much more rapid return back to the baseline and normal vision when you take the goggles off than it took to adapt to it the first time. So when I put the goggles on, it took almost a week to actually move through the world normally. When I took the goggles off, it took about a day, which tells us that the old setup in the brain is probably still there. So that was our question leading into our second experiment too. Are we making a new map of the world in our brain, and then do we get a return back to the old map? Do they both stay? And I'll take you through some data looking at that with our measurements on fMRI. So this is now the same inflated brain I showed you before. We won't have to look at many more colors in the brain, but these colors represent visual field maps, maps of visual space running off the top part of the brain here. This big circle, look at all these things labeled IPS. This is all the visual spatial part of the brain. And we made really detailed measurements of these to see if these neurons are changing their response to visual space. So this is a normal visual space, looking at the left side of the world. We want to know if they turn to the right side of the world. So visual space, I'm going to show you a lot of measurements that have responses that show contralateral, so the opposite side of the world, and we want to see if this circular response shifts to the ipsilateral side of the world. We know behaviorally they can change, but what's going on in the brain? Are these maps changing? And this is the overwhelming slide of lots and lots of measurement points of many, many maps, looking to see what parts of these maps represent the opposite side of space versus the same side of space. And this is one hemisphere and the other hemisphere, so left side of the brain, the right side of the brain. And these are all early maps up to the IPS one. There's no change. So here, number 1 would mean you're keeping your same representation. A negative one would mean all of the voxels within that area of the map have shifted their responses to the opposite side of the world. So negative 1 is completely changed. 0 is about half and half. So anything getting towards 0 is actually pretty interesting to us. On the left side of the brain, in these IPS maps, starting at map 1, shifted quite significantly to have responses over the prism adaptation period to represent the opposite side of space. So this is quite exciting for us, because what we're doing is taking an adult brain, only changing sensory input, there's no damage happening, and we're managing to make the brain recalculate what it's doing with this visual information. So this leads into ideas of how you could maybe tap into this to help with injury to the brain, things like stroke and stuff. The right side of the brain is something similar in the same maps. You see this shift, very, very same side of the brain, a slightly later shift, and we see some differences in other types of lesion studies in this region of the brain. We see differences in timing of these responses across left and right hemisphere here. So we have some theories then about what we're doing. Are we taking, like I said, the initial map, erasing it, and making a whole totally new one? Are we taking the initial map, keeping it, and adding a second one on top of it? And we can look at some of these things with our new methods, where we can look at the receptive field information. So there's also the idea of, do we have this big receptive field that seems to overlap into the same side of the world a little bit, and is dropping out signal on the normal side of the world? So right side of the brain, we look at left side of the world. What if this right side of the brain saw the left side of the world plus a little bit of the right? If we just erase the left side of the world, it would look like we're just looking at the right. Well, our question there is, so that would be, overlap this growth meridian here. We would erase all of the contralateral, opposite side input, and be left with just responses on the same side of the world. Or do we get new representations? Do we get more coverage of the same side of the world? In this case, we would see a shift into new space. And we have, that's our comparison. We look at day 14, and our subject can see how far into the same side of the world these maps represent visual space. So again, we're playing with changing the brain, we're still using maps of visual space. Yellow and white here mean coverage of visual space. This is a little 12 by 12 chunk of visual space. Center of vision is here. On day 0, this right side of the brain is looking at the left side of the world. By day 10, it kind of shrinks down. By day 14, it's expanded into new representations of visual space. So we're bringing up what probably was a really, really weak representation of visual space from birth, and really building a new, strong set of synapses that can now deal with information coming from the side of the world that's not supposed to normally deal with. And we see this again in a second subject, where we have opposite side of the world like normal, start shifting over a little earlier here, and shifts way over by 2 weeks into the experiment. So it's very exciting for us. We're creating a new map of visual space in the adult brain. Now our next question, like I said, so that was, can we create a new map? Now does the old map stay or not? So here we are on day 14, we got a new map. Behaviorally, we think the old map stays, like I said, because they can take the goggles off and start using that old representation to deal with the world within a day. If they didn't do that, it would take probably another 2 weeks to adapt back. So you can imagine all these neurons like, hey, I like the left side of the world. Oh, nope, now I like the right side of the world. Oh, now I like the left side of the world. These neurons are going, I like the left side of the world and I like the right side of the world, and keeping them both. And they're able to come back to liking the left side of the world pretty quickly. So our two models here would say over, if you lost the original map, it would take about 2 weeks to shift back, whereas if you shift back much more quickly when we take these off. So I have this comparison data now to look at. What we just saw, we shifted over to this opposite side, same side of the world, the ipsilateral side of the world here. And the maps lose this ipsilateral coverage quite quickly by day 16 to 18, depending on the subject. So we shrink back to only responding to the opposite side of the world pretty much in time for behavioral measurements, so within 1 to 3 days. All right. So we have some questions on, does the whole map change? Do certain neurons change? Are we just getting noise in our measurements? These are all things people looking at our data ask about. And what we found is that not all parts of each, so for each map, there's a section of cortex and all the neurons in it. Not all the neurons change, but some of them do. So we think that there are certain types of connections that these neurons have that allow them to bring up the representation of the wrong side of the world, basically, and then maintain that for a while. So we looked at all of them, all of the neurons and all the voxels that had representations of the wrong side of the world on day 14, and saw where they were originally. So these guys weren't just quiet that came online later and represented the new side of the world. They actually used to look at the opposite side of the world and then shifted over. So just these neurons specifically start out and ended up on the opposite side of the world like they're supposed to be. Ones that never shifted stayed unshifted. So these guys never shift over, and they stay pretty much consistent across the whole experiment. So you get a subset of the responses in this map, which makes sense because we have mapped in multiple things. You get a subset of response of the map that we think is mostly involved in this visual motor transformation, taking visual input and giving a new visual space to the motor output. So these guys are involved in doing that. All right, so my last little part is to tell you about the readaptation experiment. So these same guys came back and put the prism goggles on again. So we wanted to see if we could keep the second map as well. Does the new map stay, or does it disappear? So we have one map that's been there since childhood, and one map that came during our 2-week experiment. And then what happened to that second map? Did it just completely get erased again, or is it more quickly able to be used again in a future experiment? So these people, and by day 4 to 7, we started seeing, well, day 4 in the first experiment, we saw no ipsilateral responses coming, so no shift in the representation of the world. But what if we could see it by day 4 in a readaptation experiment? So this is 3 to 9 months later, depending on the subject. They came back. They were able to behaviorally adapt quite quickly, with much less adaptation needed. So they were much better at the maze right away, and came back to baseline much more quickly. So this is putting the prism goggles back on. They can do the maze. Note they're not up at this like really slow time up here. They're a little bit worse than baseline, but over a day they get back to normal speed of walking the maze the second time they wear the prism goggles for an extended period of time. And taking them off, they have a quicker shift back as well. So now we have questions looking at similar models. We have our experiment 1 over here. Maybe we keep the original map because it was there since childhood, but maybe the new map we made in adulthood doesn't get retained. Maybe it's just such a weak set of connections it doesn't stay around. In which case it would take another 2 weeks to build that representation set again. Now, if like we think behaviorally it stays around, it might shift back to this new representation within 4 to 5 days. And so we would look at this time point here where on day 4 prime, so experiment 2, are we in the normal side of the world still and haven't actually shifted, or have we shifted already? And we found that some of our maps actually have shifted already. So our IPS maps, the ones that shifted over, remember this is one is normal representation, negative one is not. These maps have shifted over from the first experiment. They come back, not all the way back to normal actually, and then shift back more quickly with the second set of experiments. So we have actually permanently potentially changed these subjects' brains to some degree where they have the strengthened map of the opposite side of the world for at least up to 3 to 9 months. We can look at this in these plots again. Here is our baseline, shift over to new map on day 4. No ipsilateral day 4. The first experiment shifts over about 2 weeks, shift back quickly. Second experiment, all back at contralateral side of the world. By day 4, it has shifted over quite significantly into this opposite side of the world again. So day 4 here, no data. Day 4 here, lots of data. And it shifts back to day 5. And we see this again here, where we have day 4, first experiment, no responses. Day 4 prime. Second experiment, we see lots of responses. So the second map, it's maintained in the human cortex as well. So we're able to take a normal person, change their vision, where they have to interact with this changed environment, and actually alter how their brain responds to the world in a somewhat semi-permanent fashion. This gives us hope for applying this then to other types of disease. It gives lots of information. It tells us how this brain is organized, how the brain is organized in the visual cortex. It tells us information about what regions of the brain are involved in dealing with these changes in visual motor processing. And then it lets us hope, think of ways to look at rehabilitation for patients who have damage. So what if you have stroke to one side of your brain? Can you take the other side and get it used to looking at both sides of the brain? So you could actually use both sets of maps on one side of the brain. You might be able to restore vision like this with enough training. And people do this now a little bit with these vision studies and can shift these visual fields from the opposite side of the world to a little bit of overlap, the same side of the world, with similar, really small shifting prism goggles. This would be a very drastic way to kind of reroute some of your visual processing into parts of cortex that are still alive and still functioning. We haven't tried this in a stroke patient yet, because clearly it's kind of a hazardous experiment to start with, but these are things we can start looking at. And how can we take an adult brain and make it fix it when it's been damaged? And so it gives a lot of hope for this trauma patients and stroke patients. So we're using this next then to look at things like genetics behind it, looking at people behind this, about 77 people who are these for an hour. Quite a bit of vomiting actually over the hour. But we're looking at genetics underlying brain plasticity and seeing how people differ in different types of there's certain markers for cortical plasticity, for ways brains can be organized. We're looking to see how different individuals can deal with the PRISM goggles in different ways, how they can learn motor tasks, how this divides up by genotype. Some people learn more quickly than others. Some people need more training, so this gives a way to look at patients and say, well, you're in this specific set of genes, so therefore you need, your insurance company, to cover the next year of therapy for you to get back to where you should be, versus you might only need 3 months because you're in like the rapidly adapting set of people. So we're looking at a lot of these things too, branching off from this, all from the world of initially studying visual field maps. So, my overview of the lab, some of our projects, and our fun one to look at. Thank you. Any questions for me today?
Don't be shy, there's no stupid questions.
Yes.
I'm just wondering, has this work been generalized to other areas of the brain, or, because when I'm thinking about it, it sounds like it's sort of a representation of generalized learning. So you're wearing the goggles, you learn to go left instead of go right, and it's, you know, it's mapping to that part of the brain and adjusting as such. So have people, for example, like learning to play the piano, or, you know, certain skills that you don't already know, if you replicated these studies doing other things, would you see similar results in different areas of the brain and whatnot?
I think I can answer your question in a couple different ways. So one, the genetic study we're looking at these genes specifically because they show differences in driving ability and learning to drive. And so it's a way of learning motor tasks, and people seem to differ on that genetically. And Dr. Steve Kramer here has been doing that for a while with that PRISM goggles, some of his paradigms. So people do differ in terms of actually the ones who are the most plastic, have the greatest ability to be organized, have the most problems doing motor learning, and some of the pathway may be too wildly adjusting. Like, oh my god, now it's this way, now it's this way. Imagine brains like overcompensating all the time, they have a harder time refining the skill. Now, other studies have looked at, well, one question I think you have too, is this a cognitive ability to just learn things in a sense versus visual and motor changes? And this cognitive learning, perceptual learning can play a role in this. It's different things. Learning I must go left versus right doesn't help you so much in this paradigm. It helps you a little bit. Perceptual learning, kind of like how you would learn to look at different contrasts in an image, and you can get better at discriminating two oriented lines or contrast levels, probably plays a role in this as well. I don't know the genetics behind that, how people differ exactly, but there are differences on this. And then in terms of other senses, like auditory changes, somatosensory, there's a wild controversial field about cortical plasticity, where everybody likes to claim the brain changes because they get funding and get famous. And people define plasticity, and Jacob's laughing because I rant on this in lab all the time, people define plasticity in a variety of ways, from something like this, where we call it functional plasticity, where we think we're changing the weights of little neuronal synapses, so we're strengthening things already there, versus plasticity like Christopher Reeves, who had major spinal cord injury and was quadriplegic. So regrowing spinal cord is a whole other level of plasticity. And people use the same term for everything. So we're trying to define these things really well. Visual literature has a lot of claims of plasticity, and we came in studying that and disagree with most of it. However, the somatosensory world has a lot of cortical plasticity studies where they do things like bringing fingers and children together and watch these same maps of now somatosensory, so body space change. And it looks like somatosensory cortex is much more flexible than visual cortex, for example. And this makes sense to me in terms of evolution. And you want your visual, you don't want the visual world to be changing. You want your brain to like hardwire in around the age of 5 and say, okay, this part of the world means it's this part of the world. Versus somatosensory, you get damage to your fingers and your skin a lot, and your neurons kind of take over. Well, I used to represent this finger, but now I have a little bit of information with this finger too. It's a little more flexible region of cortex. So one question we have going in is, do we have differential ability of the cortex to change across the brain in adult and in childhood? And if so, how does it differ across people? And how can we tap into this and alter it? I have like 4 different answers to your question.
Would doing this experiment benefit you after you go back to normal?
That's what they wonder when they went into it. Well, there was the worry that they would always turn the wrong way and something will drive 5 years later. UCI started a whole panel to look at our liability for this and the risk to the patients. Because of the history of behavioral studies and people recovering in the past, fine. We thought they'd be fine. Now, they have people going into it wonder if they benefit. Like, can they use information, kind of extra processing information? It's possible. We have not looked at this yet. That is kind of a next step to see if we've enhanced somehow. And And I mentioned perceptual learning. So when you get training on looking at two different oriented lines, you're basically enhancing your neurons' tuning of telling oriented lines apart. It can be very specialized, whatever you're trained up on. There's a lot of kind of sham companies to good companies out there that deal with rehabilitation and brain training or brain games, and they claim they're going to make your IQ higher, for example, and they'll train you up on a working memory task, and you'll get really good at working memory tasks. It doesn't necessarily correlate with anything else, but you get better at the task they train you on. And so it seems like here that they make better on some sort of visual motor task, but we're not sure yet. You have the next question.
I just have two questions. With the version cannot see the color, so can we use the technique to identify that?
To see the which one? Say that again.
We know that some people cannot see the color.
Oh yeah. Uh-huh.
Yeah, but they still react the same thing with the normal people when they put the display.
With this, yeah. So when they can't see color.
Yeah.
Yeah. So ones who can't see color have a problem in the retina, and they have, in some cases, depending on the type of color vision loss from birth, there's different genetics behind it, and they can have a variety of cortical compensations based on how their retina's changed, but it doesn't affect the maps themselves really in terms of how they change in this condition. So it ends up being a different processing pathway. So you have color pathways are altered and used in slightly different ways than the visual motor pathways. Interesting question.
The second question, with the people have only one eye on the right and the left, but on the images we just say the signal from right and left, but how about the distance?
Yeah, that's a very good question. So each eye has a left and a right in the visual field, right? And so you take information from each of these two eyes and go to one side, each of these to the other side. In monkey studies on this, when they look at retinal lesions, they usually cover one eye and only look at the other eye because you get overlapping responses to each eye, and it becomes complicated to work apart. We studied this in a sight recovery patient, similar, not a prison study, but a sight recovery patient who was blind from the age of 3 to 42 and had one eyeball and had scarred over the other eye completely. And he got a corneal transplant at the age of 42 and got his vision back. And we studied what happened with this. And his brain, because he's been blinded during his critical period of visual development, his brain had drastically changed. But just closing one eye and doing the study in a single eye, you get the same pattern of map presentation with slight, small variations because a different field of view than this eye. There's overlapping regions and then kind of single eye regions. But you would drive the changes from the maps in the same way as using just one eye. The depth would be different. And so without two eyes, you don't understand depth in the same way. You have to use a lot more of those like illusion cues I showed you about the table at the beginning to understand that there is depth. People can understand depth with a single eye or with misaligned eyes, but they don't see depth in the same way. They may have a different, less so. The problem with prism goggles, for us, we have to line it quite carefully in the prism so they don't get a lot of double vision. I mean, having a certain range they can see normally over and the rest is double. And with a single eye, you may not have that problem. It might actually be easier using that. Adjusting the left-right difference would be different, but adjusting the weirdness and depth, that's kind of the second problem would be. Yeah?
I read in an article that you've done some research that says that different parts of the brain perceive color differently. Can you explain more about that?
Yeah, so I think a way to think about that is there are regions of the brain that deal with color processing. And so I'll tell you about brain history. So brain history, they're like, oh, back in the Egyptian days, I'll start really far back, they viewed the brain as a mucus producer. So it moved ahead from there. But we started viewing the brain as, hey, the brain is a giant thing that does thinking. And from there, people started dividing up the brain into small and small pieces. And one of the big discoveries was in monkey that there was a region of the brain that if you knocked it out, you lost your ability to see all motion. And there's another reason if you have a stroke or damage that area of the brain, you lose your ability to see all color. And so we know that there are specific sections of the brain that involve very specific types of vision processing. So areas you knock it out, you lose your ability to understand faces. You can't see faces anymore. So people can have lesions and have this happen. So people have been excited about this and study them in detail to see what the differences are. Now, from the very, very modular view, we went from mucus ball, giant brain, one thing, to little tiny modules. We're kind of coming back to the idea that it's a network, which makes sense. There's a lot of processing going on in pathways that have several stages of processing. They all talk to each other, but there may be key nodes in these networks that if you lesion them, you lose your ability to see color and stuff. And surprisingly, the color area has had a lot of argument about where it is and how exactly we are consciously aware of color. We know a whole lot about color in the retina and how it comes into early cortex, and then we go, and then we understand color. So there's very, very good circuits drawn and how we bring in differences in the photoreceptors in your retina have different responses, that we get this difference between responses to figure out what the color wavelength ranges, and then understanding our conscious perception from that happens somewhere forward of that in the brain. So we're coming into the back of the brain in V1, we do a lot of color computations, and then we have a region that, if you lesion it, you lose your idea of color, but we're not entirely sure how that makes a computation understand color. And so we were, in the pilot you saw, we were looking at measuring these maps and seeing, instead of getting a blob of color responses across the bottom part of the brain, we were dividing up into specific visual field maps. We could try to ask the question of how do these maps differ in different color processing. So we have kind of several little chunks of maps to do color processing. If you get a lesion on the top of the brain, you don't lose color vision, like ever. So it tells us there is some difference between these pathways.
Oh, and I also had a question. I've heard that, or I've read that, like, the right side of the brain is more, like, able to deal with, like, art and, like words, and the left side is more like science and math. Is there a connection with, like, your like visual, how you see the world, that connects to like more intellectual, that part of your brain? Or
So I got to go to textbooks and say that all textbooks are 10 to 20 years out of date, period. Okay, ask any science professor, it takes 10 to 20 years for information in textbooks. Two things. One, we don't use just 10% of our brain. That's wrong. We use our whole brain. Two, laterality is not as simplistic as what everyone is told. So the creative side versus math side is not accurate, period. The visual sides may start to differ when you get into color processing. We maybe see a little bit of differences. Language, which they say language is on the left. Actually, no, language is on both sides for most of it, and a little bit extra on the left. So there are distinct behaviors that appear to have this laterality effect we don't fully understand. And people may get left damage and lose speaking ability, but they don't lose language comprehension necessarily, or other elements of language. And singing may be preserved on the right more. So there are some distinctions, but there's not the distinction that textbooks like to say, this is the more creative art side, this is more language math side. And people are trying to backtrack to see exactly what measurements make us think this. In some cases, it's kind of odd damage. The lesions, when you study a lesion, it means you have a big area of damage to the brain that's uncontrolled in human. When cut out someone's brain, usually that's like certain conditions, like epilepsy allow for that. But so we have these kind of uncontrolled lesions, and people have made big claims based on some of these that now are being refuted a lot more. So you don't see this so much in vision. And in fact, in color image, you could color vision. If you got damage to a really specific map on either side of the brain, you could lose just color in that little chunk of the world. So people tend to not notice it because you move your eyes around and compensate a lot. But you could use one little quadrant of color to stay really fixated. And someone asked you about colors in the space, you wouldn't actually be saying it. So you don't get that much lateralization in vision until maybe higher up. There's some question about face lateralization, maybe more possibly than one side than the other. That does differ between people, and we don't fully understand the breakdown yet. Yeah.
With the, it's just example in my head right now. Example, we have the people wear the goggle, so they invert the word, right?
Mm-hmm.
It's technique experiment, but their brains, the rotation, they don't connect. They don't have rotations. This means left to the left side, right to the right side. So how can it affect? This means they see the world like a normal person, or
So like the split brain patients?
Yeah.
So a patient with really bad epilepsy, they tend to cut the connection between the two lobes so the epilepsy, the seizures don't spread to the opposite side of the world. And at first I thought there was no issue with this. And then they started testing it in more detail and discovered that here is one of the things, if you get information on the right side of the brain goes to the left side of the world, and the left side of the brain goes to the right side of the world, and they don't talk to each other, you can do weird things where you can ask them to what they saw, and speech is more heavily concentrated on the left in most people. So if they see something that goes, so speech on the left, they see stuff on the left side of the world that goes only to the right side of the brain, they'll tell you they didn't see anything, but they can write down that they saw something. So there's videos of it. So you search split brain patient on YouTube, there's some great videos of these patients doing this, and they're like, oh, I didn't see anything. And I go, what'd you draw? And they're like, oh, I drew a frying pan. And the frying pan was, and they have special setups where they really restrict vision. And so you're asking, like, what happens with this, right? Because we assume with this that these show some underlying connections between the brain, and we're not growing new ones, but they're kind of weak ones that we're using for this. So if we flipped it, I don't think they'd be able to adapt to this very well. I think we have to have crosstalk between the hemispheres to drive this type of, not growth like axons, but kind of this change in strength of response. So yeah, look up split brain patients on YouTube. It's very, very interesting. So this gives us some idea that there is lateralization in certain elements that doesn't hold up in all things that we study. So people are trying to figure out how that plays a role in science. Oh, yeah?
Is there like a reason why, like, you know, like the left side of the brain controls the right side of the body and all that? What is it called, desiccation of the nerves or something?
Yes, the desiccation of the pyramids, the motor and sensory stuff comes outside the body here. No, there's theories of evolution about why this might have developed. So I don't have a good answer. People have a lot of theories about it twisted or this happened. It could have been random. And so when we look at ourselves, I think about evolution a lot for how we got to where we are and how we compare to monkeys, because there's all this monkey literature that I had a big fight in grad school with color regions where they were saying, oh, they're different in human than monkey. That can't be. I'm like, we're 25, 27 million years different from this monkey, so something can change over that time. We're also not at the endpoint of evolution, and things happen by random chance. So in random chance worlds, we randomly could have crossed one point and it worked, so we stayed. There are some animals and some things that don't cross necessarily, that I like invertebrates and things, they don't necessarily cross. And there seems to be no disadvantage to this, but when people ask me why is the forebrain up here versus back here, well, it's a newer part of the brain, so it kind of grew on top of the old parts. So we use what machinery we had and grew more parts to it. But yeah, I don't know why, I've always wanted, it seems so weird, right? Like, if you get damage up here and you use this side, I don't know what advantage that would have.
Do you feel about the whole, like, left-handed versus right-handed debate? Like, why is left-handed, like, more violent than right-handed people?
Why is it what? More?
Why is it, like, less dominant?
Yeah, I don't know. It's an interesting debate, and we just did a study with genetics prisons, and we used only right-handed people because we didn't know how different it would be, and we had a debate on everyone. The lefties on the program, all the researchers were upset. But we had to be consistent, and there's not a lot of understanding yet about what makes brain dominance exactly. We know that one thing that can make you switch is damage to the brain, especially in utero. If you're left-handed, I'm not saying you have brain damage in utero, but in some cases you might have. Or you might be right-handed and used to be left-handed, so it's hard to tell which way your brain happened. As a child, if you damaged the left side of your brain in motor areas, you would switch over to using the opposite hand. So you switch handedness depending which side of the brain you damage. So the left side controls right hand. If you damage your left hand during early childhood, left hand representation, damage the hand representation left side of the brain that controls your right hand, you would then be able to, in this more plastic time, be able to use your left hand instead and be completely left-handed. So some theories are maybe that happens really early on in some cases. Left-handedness also runs in families, which gives us a genetic component. So we have some genetics for how the body forms and twists during development, and in some cases the brain may just twist one way versus another. People can have parts from the wrong side of the body or the opposite side of the body. We can have entirely internal organs flipped, and maybe something similar happens in brain development and just how we trigger what this dominance is. And left hand, so one thing people look at to distinguish left-handed people who may have had, say, damage, really subtle damage early on, that some have the right time in development in utero caused dominance to switch and no other issues. Well, they may have language on the left still and be left-handed, whereas some people who may be left-handed and never had any damage may have language on the right side of the brain. So the brains are so what we know about laterality has switched in many left-handed people. And so people are playing with genetics versus really subtle damage. My son's left-handed, I don't think he's damaged. He's just fine. But there is also more ambidextrousness that comes up with a lot of left-handed people, but it's hard to differentiate, even to this day, how much of that is school playing a role in saying you must be right-handed, or here's, 90% of the kids using the right hand. My son learned to write late because he kept trying to use the right hand because all his friends did. So by 5 you could write with the right hand, with his left hand just fine, but at age 4 he was still pretty sure he should use his right hand because Scott over here uses his right hand. So it's hard to pull apart these early social interventions and teaching interventions from some of this too. So it makes it really complicated. And when you go in expecting one area of the brain to be on the left instead of on the right, people sometimes restrict down to right-handed patients or subjects.
Which makes like a whole field for left-handed people.
Right, I know. We have a lot of left-handed people in the lab too, so maybe we'll do a little study sometime. Really look at the difference.
Any other questions? All right, how many people here are RSVP for the lunch? Okay, you guys, we're going to get together and walk